Good morning, I'm Laura Canesi. I'm a professor of physiology at the Genoa University. Today I will talk to you about our data on environmental physiology, data that we share with the Sorbonne University, the Laboratory of Developmental Biology in Villefranche, on neuroendocrine control of early larval development in a changing environment, utilizing the model of marine invertebrate Mytilis galloprovincialis. What is environmental physiology? Environmental physiology studies the responses of an organism to environmental changes, and in this way it's closely related to comparative physiology and also to evolutionary physiology. But the environmental physiology approach is especially relevant in the context of ongoing global changes, as organisms are exposed to increasingly altered environment by human activities. And there are several reviews, also a few years ago, on this subject. I would like to remember the Keystone review of George Somero on comparative physiology and global change. What is the relationship between physiology, climate change, and health? Also, according to the Physiological Society, physiologists are playing a key role in delivering on climate change's mitigation and adaptation goals. Physiological research is helping the fight against climate change. But this not only applies to environmental physiology for human health, but also to animal health, especially for those animals that live in the ocean, because ocean ecosystems are particularly affected by climate changes. As stated by the United Nations, we are now facing a triple planetary crisis. This refers to the three main interlinked issues of climate change, pollution, and also biodiversity loss. But such a crisis is particularly evident in the oceans, with potential consequences and impacts on human health. Because, for example, the impact of pollution, of ocean pollution on human health is now recognized. This is a summary of the report of the United Nations, the IPCC 2019 report, where the main environmental changes occurring in the oceans are depicted. And I would like to remember just the ocean heat content, so the temperature increase in the ocean, the decrease in pH, ocean acidification, and also the depletion of oxygen. Invertebrate models can represent suitable models for investigating physiological responses of marine species to global changes. Why? Because invertebrates represent 95% of all animal species, and mollusca, for example, represents the second largest phylum and the first in the sea. Marine invertebrates also represent, with the different groups, key components of marine ecosystems. But invertebrate models, and in particular mollusks, have been long utilized in physiological research. You all will remember the example of the giant squid axon or the Aplysia model in sensitization. We utilize for environmental research the model of bivalve mollusks. Why? Because marine bivalves live in intertidal and estuarine coastal areas. They are naturally subjected to continuing fluctuation of different environmental parameters, and thus they are potentially more vulnerable to further ongoing environmental changes. But in order to survive to these complex environments, bivalves need efficient physiological mechanisms to respond to the number of abiotic and biotic stressors. But what we know of the physiological control of function in bivalve mollusks? As most invertebrates, bivalves lack a proper canonical endocrine system. However, a neurosecretory and neuroendocrine system appear very early in evolution, so all metazoans, and thus all invertebrates, possess neuroendocrine cells releasing signaling molecules in their body fluids. This implies that in bivalves, as in most invertebrates, the modulation of physiological function is essentially neuroendocrine. And although information on bivalve neuroendocrine system is largely still descriptive, in adult mussels, we know neuroendocrine responses to stress that have been described in detail, in which the monoaminergic system plays a central role. If you look at this picture, Different environmental stressors affect the functioning of bivalve ganglia that release a wide range of signaling molecules, including monoamines, that travel in the hemolymph, in the body fluids, and reach target cells and tissues, thus orchestrating the response to environmental stress. But what is the role of monoamines in particular on bivalve physiology? In adult bivalves, monoamines play multiple roles in several physiological processes. Once released by the ganglia, and once the monoamine level changes in body fluids, they can affect immune cells in the hemolymph, with consequences on immunomodulation, or directly target different tissues. For example, they control ciliary beating at the gills, that are key players in feeding and respiration. They regulate muscle and heart contraction, and also act on the gonad, affecting reproduction, growth, and also spawning. But what do we know on the monoaminergic system in bivalves? Thanks to the recently published genomes of some bivalve species, some components of the monoaminergic system have been identified. But we did not concentrate on adult muscles, we concentrated on early bivalve development. Because climate change and pollution can affect the organism's capacity to maintain homeostasis, but in this way they can affect individual health in terms of differing function. This is particularly important for early life stages that are more sensitive in all species to environmental changes. And here, environmental physiology meets developmental physiology, because investigating the effects of environmental changes and pollutants on physiological mechanisms of development can provide useful information in predicting the impact of these changes on more vulnerable life stages of key invertebrate species and the possible consequences on biodiversity. Here is a scheme representing the life cycle of bivalve mollusks. As you can see, they have external fertilization in the sea, they are gonochoric, and once released in the water, fecundation occurs from gametes, and the first free-swimming larva is the trochophora. Then the trochophora develops into the diveliger stage, which is the first shelled larva. And the diveliger, in turn, grows and develops until metamorphosis and giving rise to the adult. But it's interesting that within 2 days post-fertilization, from the trochophora to the first diveliger, the key processes of shell biogenesis and neurogenesis occur. Information of neuroendocrine control of early larval development in bivalves comes from a key study of a few years ago on Castorsa gigas, the oyster. The author demonstrated that serotonin and dopamine control initial shell formation by regulating the expression of two key genes, tyrosinase and chitinase, that ensure organic matrix deposition and remodeling. But exposure or oyster larvae, the trochophora, to ocean acidification suppresses the biosynthesis of serotonin and dopamine and their related signaling pathway to the TGF-beta SMAT. This would subvert the expression pattern of both tyrosinase and chitinase and cause the failure of shell formation in early D-shaped larvae. And in the oyster, in this way, they demonstrated that both serotonin and dopamine are pivotal for shell biogenesis. But oysters are not a key species in the Mediterranean. And according to the UNEP map, the Mediterranean Sea represents a hot spot for global changes. It's a closed sea. So we focus on the utilization of another bivalve model, the Mediterranean mussel Mytilis galloprovincialis, for bivalve larval development. First of all, here is the species, Mytilis galloprovincialis. We focused on development of the shell, so we studied the time curve of shell biogenesis. We made use of double staining with chalcofluor for organic matrix deposition and calcine for calcification. As you can see here, from the trochophora at 24-hour post-fertilization, shell deposition in the organic matrix begins in blue and proceeds through development. Later on, calcification begins. At 48 hours, we have the perfect diveliger larva with a calcified shell. But the organic matrix is important because it represents the blueprint for cell calcification. We follow gene expression across these larval stages, and we observe a strong upregulation of genes involved in matrix synthesis and remodeling, in calcification genes, and a large increase, a large upregulation of both serotonin and dopamine receptors. And pharmacological evidence provided the further cues because, for example, inhibition of tyrosinase and blockage of the dopaminergic neurons prevented normal organic matrix deposition, calcification, and normal shell formation. For example, inhibition of tyrosinase inhibited by 80% the shell matrix deposition, and inhibition of dopamine receptors resulted in more than 80% malformed or arrested larvae. Then we follow the onset of serotonin and dopamine system across early developmental stages. First, the serotonergic system, and we observed the appearance of the first serotonin immunoreactive cells in the trochophora at 24-hour post-fertilization. As development proceeds, the number of serotonergic neurons increase up to 7, with the appearance of the first tiny neurites. As to the dopaminergic system, we don't have specific antibodies for dopamine, so we made use of in situ hybridization to follow the expression and localization in the larva of components of dopamine synthesis, catabolism, and dopamine receptors. And we observed that dopaminergic components are expressed from about 28-hour post-fertilization with an apparent delay with respect to serotonergic cells. But to have more information, we made use of data from the recently published reference larval transcriptome in Mytilis galloprovincialis. And so we followed the quantitative expression of different serotonin and dopamine components. As you can see here, serotonin components peaked in the gastrula stage from 12-hour post-fertilization, whereas the dopaminergic system peaks later. Overall, exploring all components of all monoamines, we can conclude that the monoaminergic system of muscle larvae is composed of 6 circuits: serotonin, dopamine, noradrenaline, octopamine, tyramine, and histamine. But serotonin and dopamine are predominant in the first early larval stages. Then we checked the appearance of serotonin and dopamine-producing neurons by HCR, in situ hybridization chain reaction, and we observed a distinct time course of serotonin-producing cells and dopamine-producing cells, and observed their neuronal localization. So serotonin-producing cells appear from the gastrula stage and proceed, and are located in the apical region of the larva, whereas dopamine-producing cells appear later on from the trochophore and localize in the ventral neurons of the larva. But we know that in addition to neuroendocrine control, different environmental cues, biotic and abiotic, can affect Mytilus early development, as observed in oyster larvae with acidification. We first check biotic factors, and the main biotic factor is the presence of bacteria. There are plenty of bacterial species typical of the marine environment. We also check the composition of the bacterial community in eggs, sperm, and in the developing embryo, where Vibrio represents a significant component of larval microbiota. However, different marine bacteria affected to a different degree normal larval development, and in particular those Vibrio species that represent recognized oyster pathogen. And for example, Vibrio tasmaniensis, which is a recognized oyster pathogen, was active from very, very low concentrations. Among abiotic factors, acidification, the Villefranche group carried on some studies on the effect of acidification on muscle larval development to compare with the oyster, and they observed that, again, acidification affects gene expression, in particular those involved in shell formation and in stress response. In the Genoa University at the Environmental Physiology Lab, we investigated the effect of temperature alone and in combination with bacteria in 2 experiments. We evaluated larval development, mortality, and gene expression. And when we look at the phenotypes and survival, temperature alone just 2 degrees of the temperature increase, these values are those predicted with the temperature increase in the Mediterranean in the next few decades, are sufficient to strongly inhibit normal larval development, but no effect on larval survival. In the second experiments, combined effects of temperature and bacterial charge resulted in a stronger effect on larval development and also in mortality at temperatures of 20 and 22 degrees with respect to the control of 18. When you look at gene expression, temperature alone affected the time course of gene expression across development, and the most affected genes were those involved in shell formation, antioxidant defense, ceramide metabolism, but also the serotonin receptor and immune-related genes were affected, not the dopamine receptor. In the combined exposure experiment, although statistical analysis underlined that temperature is the main environmental driver, the effect of temperature and bacterial challenges were interactive, also in gene expression, and some genes that were not affected by the increases of temperature alone were up or down regulating, like the dopamine receptor. Then we started looking at the potential effect of emerging contaminant. What is an emerging contaminant? According to the Environmental Protection Agency, an emerging contaminant is a chemical or material characterized by perceived potential or real threat to human and environmental health, or lack of published health standards. And we show that in muscle larvae, data on exposure to different emerging contaminants can provide further evidence for the monoaminergic control of shell biogenesis. We first started with the plastic additives, that are compounds intentionally added to the plastic polymer to confer the final product its specific requirements. They include a wide range of compounds from the plasticizer, flame retardant, stabilizer, antioxidant, and so on. But since most additives are not bound to the polymer structure, in plastic debris dispersed in the ocean, they can migrate to the seawater. During aging, weathering, UV irradiation are different factors affecting the release of these compounds. But this represents a complex risk of exposure of these mixtures of compounds to wildlife and humans. We first tested the effect of bisphenol A, which is a classical plastic additive, but also recognized as a weak estrogen and endocrine disruptor in mammalian system and invertebrate system. We evaluated the effects on shell formation and on the serotonergic system, and we observed alteration of larval phenotypes, altered expression of tyrosinase and OX, a key developmental gene, upregulation of tyrosinase and downregulation of serotonin receptor, and a decrease in the number of serotonin producing cells. Its conjurant, tetrabromobisphenol A, TBPA, is a flame retardant and a thyromimetic compound in mammalian system. TBPA affected different neurotransmitters, serotonin, dopamine, and also GABA. These effects were accompanied again by altered transcription of shell-related genes in the alteration of organic matrix deposition and calcification, resulting in larval malformation or arrested development. So, with BPA, that represent neurodevelopmental disruptors in muscle larvae affecting different neurotransmitters, we could design a potential adverse outcome pathway that starts from the molecular initiating event, altered synthesis of neurotransmitters, to cellular, tissue, and organism effects that can and could affect larval populations. We also tested the biocide triclosan, which also affects larval development, but the effect here is specific on calcification. So matrix deposition in the shell was not affected, and the monoaminergic system were not affected. In the last few years, we concentrated on another class of emerging contaminants, environmental pharmaceuticals. Pharmaceuticals, given their increased consumption, low retention in wastewater treatment plants, and constant input, are widely detected in all marine coastal areas and thus are considered as pseudo-persistent contaminants. But pharmaceuticals are designed to act on specific molecular targets in mammalian system at very, very low concentration. So their presence in the marine environment can represent a threat to non-target aquatic organisms that can be continually exposed to these compounds, although a very, very low concentration, over the whole life cycle, including early life stages. We first tested the effect of a common anti-inflammatory drug, diclofenac. Diclofenac induces shelf malformation from very, very low concentration, nanogram per liter, and affected the transcription of shell-related genes, biotransformation genes, and serotonin receptors. Then we moved to other commonly detected pharmaceuticals with different function, carbamazepine, an anti-epileptic drug, propranolol, an antihypertensive drug. And both compounds altered larval development, again a very low nanogram per liter concentration, with an induced distinct impactor of gene expression, including alteration of shell-related genes and neuroendocrine-related genes. But these widespread pharmaceuticals do not specifically target monoaminergic components. In contrast, antidepressants, that represent one of the most utilized pharmaceuticals worldwide, are specifically designed to interfere with components of monoaminergic system, potentially affecting non-target organisms in the sea. Several biological effects of antidepressants have been described in a wide variety of marine species, from molecular and biochemical effects, histopathological changes, physiological responses, and behavioral responses. They are frequently detected in both freshwater and marine environments, and due to the strong target conservation in the synapses of vertebrates, they have been shown to represent a significant risk for marine species, fish in particular, at very, very low concentrations. In particular, selective serotonin reuptake inhibitors, SSRI, specifically target serotonin transporters, SERT, reducing serotonin uptake and thus increasing its concentration at the synaptic cleft. A model compound is fluoxetine that blocks reuptake of serotonin in the presynaptic membrane. The consumption of SSRI has enormously increased during the post-pandemics, so their concentration, due to the low retention and degradation in water treatment plants, has increased in coastal areas, representing a potential risk of exposure to a wide variety of marine species. We know that in adult bivalves, exposure to different SSRI induce a variety of responses at extremely low environmental levels. And also for these compounds in the adult, an AOP could be designed. But when we tested the effects of SSRI on larvae, we had a surprising response. We tested fluoxetine, citalopram, sertraline, and in comparison also venlafaxine, which is the commonest serotonin norepinephrine reuptake inhibitor. It is slightly less sensitive toward the serotonin transporter. We observed significant but small effects on larval development, and not at nanogram concentration, at higher concentration, microgram per liter. In parallel, we observed significant effects in the number of serotonin-producing cells, but again, the effect was small, as shown here by HCR or TPH, the synthesizing enzyme. So we looked for the presence of SSRI targets in early larval stages. And also here we made use of data from the Mytilis reference larval transcriptome, and we followed and quantified the expression of the two SERT transporters we found, SERT1-like and SERT2-like, and compared the expression of these transporters with those of the enzyme responsible for serotonin synthesis. Again, here there is a delay in expression of SERT with respect to serotonin synthesis. And when we look with the ACR, with triple ACR, at the expression and localization of SERT1, SERT2, and TPH, we show that only at 48 hours in the D veliger, all components are expressed, confirming that the serotoninergic system is completely established only in the D shell larva at 48 hours post-fertilization. So the late expression of SERT transcript in developing embryos could explain the limited effects of SSRI on larval development. And the question now is how flexible or modulable is the monoaminergic system muscle larvae? Because from comparative anatomy studies, we know that in gastropod mollusks, the serotonin gradient is crucial for normal development of neurons. Disruption of this serotonin gradient by administration of exogenous serotonin resulted in irreversible malformation of the forming ganglia and larval death. So we tried with exogenous monoamines, serotonin and dopamine, and their inhibitors. And we observed that normal larval development is extremely sensitive to exogenous serotonin. When you look at the number of serotonin-producing cells by ACR, we observe that, like and much more than SSRI, exogenous serotonin decreases the number of serotonin-producing cells. And this indicates that an excess of serotonin can exert a negative feedback on serotonin synthesis. Overall, this data provides some molecular evidence that also in Mytilus, the monoaminergic system can support the capacity to respond to changing environmental conditions and creating the molecular and biochemical basis for adaptive plasticity in development. So, to conclude, from these studies, we can learn that responses to environmental stressor can help revealing the basic mechanism of neuroendocrine control of early development in bivalve mollusks, and that this knowledge can be used to identify early signs of impact of different environmental stressors and the molecular targets involved. So, in this slide, the environmental physiology approach can contribute to a better understanding of the potential perturbation of developmental processes of key marine invertebrates and consequences on biodiversity in a global change scenario. But before leaving you, my take-home message comes from a recent study published in PLOS Biology from the Frank Meltzner group. The title is clear: The Silent Loss of Cell Physiology Hampers Marine Bioscience. Because the author underlined that in the past few decades, the omic revolution has enabled astonishing advances in marine science. But today, genomic and transcriptomic data alone, together with ecological data, are increasingly being used to understand and even to predict the responses of marine species to ongoing environmental changes. However, in the author's opinion, which I share, the omics approach suffers from two main strong limitations. First of all, we do not know the identity of the relevant genes or the function of the coded proteins in most marine species. What is more, GO terms or KEGG pathways have little relevance for studies on most marine animals, since these databases are largely built on models, mammalian vertebrates, or model invertebrates like C. elegans or D. melanogaster. They do not possess the physiological traits of the species we are generally interested in evaluating. So the few studies in which researchers have successfully linked the genomic targets to specific cellular processes share a common feature. They benefited from decades of foundational research on cellular biochemistry and physiology that grounded their analysis. So our wish is that only with a strong community of environmental and comparative physiologists we will be able to understand how most of marine animal species work and how they respond to environmental changes. I thank you very much for your attention, and also thank my collaborators in the Environmental Physiology Lab in Genoa and the collaborators in the Laboratory of Developmental Biology in Villefranche. Thank you very much.